This video is learning targets 11 through 13 of the genetics unit. We'll be talking primarily about the different types of Punnett squares that we can use to show genetic probabilities. So what is probability? Probability is just the likelihood that something will occur. And so we use probability a lot with things like dice and cards or whatever, but probability is important in figuring out possible allele combinations in genetics as well. So if you remember, we talked about the law of segregation. The law of segregation said that the two gametes can have an equal chance of being inherited, so that if a person has a dominant allele and a negative or a recessive allele, each of those alleles have an equal chance of being inherited because those gametes or those alleles separate during gamete formation. And so here's a picture of chromosomes that demonstrate this. This is sex chromosomes, the X and Y chromosomes. A male or a female has two X chromosomes. A male has an X and Y chromosome. And so in order to show those different allele combinations, we could draw some kind of crazy looking arrow configuration like this, saying, well, this X could meet up with this X, or this X could meet up with this Y, or this X could meet up with this X, and this X could meet up with this X and Y, and so forth, and we could get all convoluted here. However, it'd be easier just to show this in something like this. A nice, neat Punnett square where you could have XX, XY, and you could use this to show that there's a 50% chance that two people would have a boy and two people would have a girl. And so that's why we use Punnett squares. We use Punnett squares to show probability, to show how probabilities can be demonstrated. This Punnett square here does not say that this couple is going to have 4 children. It says that there's a 50% chance that if they have a child, it will be a boy. Or there's a 50% that if they have a girl or have a child, it will be a girl. And so going back to our tall and short paradigm with P's, whereas dominant is tall, recessive is short. If this is two heterozygous individuals crossed here, you can see that there is a 25% chance, or a 1 in 4 chance, that that individual will be heterozygous recessive or homozygous recessive for short. We call this type of cross, where there's just one trait, tall, short here, the plant height being the trait, we call that a monohybrid cross. One hybrid is being made, one mix between those two. And so we can do lots of different crosses this way. Typically, let's go back to the one we were just at here. Usually the first thing that I do when I'm given a problem is I'll try to just establish, okay, what is the genotype of the two organisms that I've been given? And let's just say that this is a cross between two heterozygous organisms, like I've got written here. And I'll separate them with blue and red. That'll be the easiest way to do this. We're going to put the blue one on the left. You could easily put it on the top. It doesn't matter. And then to show that each allele has an equal chance of being inherited, we're going to put one allele here and one allele here, representing that each one of these boxes is going to have one of these blue alleles in it. Well, we'll put the red one on top, same idea. And again, each allele has an equal chance of being inherited. And so we're going to show that just like that. And a good rule of thumb that I like to use, because we'll be getting into Punnett squares that have multiple letters in them, and I like to group my letters. You can decide if you want them in order, alphabetical, or whatever. But if I'm given letters, I like to group them T's and A's and B's and C's and everything together. And then I like to put my capital letters in front of my lowercase letters. Just a good rule of thumb to help you to organize your thoughts here. Let's do one more practice. Let's say that there's a cross between a homozygous recessive plant, homozygous recessive for short, so that would be homozygous, same, recessive, both lowercase, so recessive trait, with a heterozygous individual, which would be different. So one lower or one uppercase, one lowercase, having one dominant allele and one recessive allele. And we'll do this again. So we'll put our little t on the left hand side, again not mattering where it goes. And we'll do this one along the top. Now, what I might ask you to do, and what you should be able to do, is if I give you a problem and you've, like the one I just gave you, and you solve the Punnett square, that's all well and good. But what I would like for you to do is be able to give me both the genotype and phenotype frequencies. So what do I mean by that? Well, genotype is the actual genetic makeup. And this is typically designated in letters, like you see here, big T's, little t's. An organism can be big T, big T, big T, little t, little t, little t. Or you can just simply say homozygous dominant, heterozygous, homozygous recessive. These are all genotypes. You cannot go up to a person and look at their eyes and see that they're homozygous recessive for eye color. You can, but you see blue. You don't see two little A's. And so genotype is what you, or genotype is not what you see. Genotype is their genetic makeup. Phenotype, on the other hand, is their physical characteristics. Notice I underlined the pH there. Physical characters. So blue eyes, brown hair, tall, short, whatever. These are the things that you see. So if I'm given this problem that I just had here, and I ask you to give me the genotype and phenotype frequencies of this particular problem, then what would I do? Well, I would start by doing the genotypes, as I usually do. And I would look at this and I would say that, well, one of the genotypes that's present is big T, little t, or heterozygous, and that there are 1, 2 of those out of the total 4. You can go ahead and reduce that if you want to. You can put it in a percentage if you want to. However, I'm completely content with 2 out of 4. And what about the next? Well, there's also a little t, and that takes them all up. That's again 2 out of 4. And so these are genotypes. They add up to be 4 out of 4. We don't need any more. You don't need to do this because this is not here. There's no need to do that. No need to do that. We don't see it here. What about phenotypes? Well, we have 2 phenotypes, tall and short. We have tall. How many of these organisms are tall? Well, how do you find tall? Look for a big T. There's 2 of them. 2 out of 4 are tall. And so then 2 out of 4 are short. And so that's it for the monohybrid cross. They don't get too much more difficult than that. All right, so here's just another picture showing you could take this one if you wanted to and do those same genotypic and phenotypic percents. Take some time to do that. However, I'm not going to for the video. So next I'm going to teach the law of independent assortment. And the law of independent assortment says this, or Mendel's principle of independent assortment, you'll also see it written as, this says that individual traits are inherited independent of one another. And so so far we've only been dealing with one trait, so now we're going to be looking at crosses with 2 different traits, like a short pea plant with yellow seeds, passing both the genes for yellow and short. And we're going to look at crosses that have to do with seed color and plant height, for instance. You could easily make this to be horses' color and their way they run, which I'll give you a practice problem that has that on there, or eye color and hair color. There's lots of different combinations you could do this with. The point is that we're going to be dealing with 2 different traits. Mendel saw in the labs that he did that traits were inherited independent of one another, meaning that when a pea plant was made with green seeds, it wasn't necessarily always tall. could have a short pea plant with green seeds or a tall pea plant with green seeds. And so this picture kind of shows that with the yellow and green and round and wrinkly seeds. We're going to go tall short because we've been there for a while. And so let's see short, tall, and then let's use yellow, green. All right. What are some different combinations between these two traits that we could have? Well, we could have tall yellow, we could have tall green, we could have short yellow. You get the idea, short green. Well, what about a cross showing this? Well, again, don't forget that we're just talking about showing what's going on in meiosis. And so we're showing homologous pairs separating in anaphase 1, and then sister chromatids separating again in anaphase 2. And so what the law of independent assortment says is that during that anaphase 1, when homologous pairs are separating, in this case, where we have G's and Y's, you could have the big G and the big Y go together, and little g and little y go together, but it could separate to where you could have big G, little y go together, or little g, big Y go together. And so you could have multiple different combinations making multiple different kinds of gametes. Remember we talked about how meiosis makes 4 gametes, each genetically different from one another? Well, this law of independent assortment is one of the reasons that that's true. And so let's take across. Let's take this one to start with. And notice that this individual, and we're going to use, say what we're going to get A and B here. We're going to say that A is tall, and that little a is short, and that big B is green, and that little b is yellow. And so what we have here is we have an individual that is heterozygous for tall and heterozygous for green crossed with an individual that is heterozygous tall and heterozygous green. And so how can we show that? Well, we're going to need more than 4 squares to do that. We're actually going to need this one, kind of big. So up the top, I'm going to write our cross again. All right, so how do we know what to put on the left and on the right? And I'll show you the mistake that often gets made here. This is the mistake that often gets made here. People do this and this. And so what's the problem with this? Well, you start filling this in, and then you're going to quickly see that only a small portion of these individuals are actually going to get both the alleles for height and for seed color. This individual, for instance, is only going to be tall. It's not going to have a seed color. That's not right. And so what we need to demonstrate is that each parent is passing all of the alleles, one for plant height and one for plant color, or seed color in this case. And so how do we do that? Well, back here when we did this little exercise, this tall, yellow, green, that sort of thing, we can do the same thing here. Well, what combinations can this red individual make? Well, it can make a tall green, right? Because it's got the uppercase. It can make the tall yellow because it's got a big A and then a little b. It can make a short little a green, big B. And it can make a short little a yellow, little b. It can make all those combinations. Well, it would get really tiresome writing those words over and over again in all 16 of these squares. And so what we're going to do to represent them, instead of writing tall green, we're going to write big A, big B. And instead of tall yellow, we're going to write, and then you'd be using red here. Instead of tall yellow, we're going to write big A, little b. And you get the idea. We're going to do this for all of them. Little a, big B, little a, little b. Well, what is the blue? Well, notice the blue has the exact same genotype as its partner, and so we're going to do the same thing on the top. And so when we start filling these in, what's it going to look like? Well, each possible offspring is going to have four alleles just like their parents. Not exactly like their parents, but they're going to have four alleles similar to their parents. And so we can spend some time filling these in. And I will caution you, as I'm filling these in, it is real easy to make simple mistakes while you're doing this. And for me, I have done 1,000 of these if I've done one. However, for most of you, that will not be the case. And so I would strongly advise caution when filling these in, because it is very easy to make a mistake. Now, most of the time, which I'll show you an example, we are not going to need all 16 squares, but in this case, we do. And so what we want to do next is let's start doing the genotype and phenotype frequencies for this particular cross. Well, how many of these do we have? Big A, big B, or big A, big A, big B, big B. We have 1 out of 16. And then we can just keep going. There are 2 of these, 2 of 16. Again, 2 of these. And there's going to be 4 of these. I can start marking them off when I find them here. This all kind of follows a nice little pattern. Now, again, handwriting's a little crazy, but you kind of get the idea. There are 9 different genotypes in this cross between 2 heterozygous traits, or heterozygous for both traits, individuals. What about phenotypes? Well, there's only going to be 4 combinations here, so it's a little bit of a break. Well, what is the phenotypes? Now, you may well be tempted to go tall, short, green, yellow. However, we have to go tall, green as one option. What would those be? Well, there's one. There's why? Because they have a big A and a big B, right? 1, 2, 3, 4, 5, 6, 7, 8, 9. What about tall yellow? Well, tall yellow, save you the suspense. There's going to be 1, 2, 3 out of 16. And then short green, that you could already tell me. There's going to be 3 out of 16 of those. And then lastly, all recessive short yellow, 1 of 16. Now, again, I won't ever have, you probably won't ever have to do a problem like this for me this big. Most of the time for me, you're going to see something that looks like this. An individual that has some homozygous traits and some heterozygous traits kind of mixed in. And so what would we do with something like this? Well, let's just take another, borrow this square here. All right. And so same idea, red and blue. Now, what kind of combinations can we get with a red one? Well, we can get again, we can get a tall yellow, and then, well, we could get another tall yellow because there's two little bees, right? We could get a short yellow, and then again, because there's two little bees, another short yellow. Now, notice that's redundant. And so on the front end, because these two rows are going to be the same, and these two rows are going to be the same. And so what we can do on the front end is actually save ourselves some work and just use half the squares. So we don't always need to use all 16 squares. Same thing with the blue one. Well, we have big A, big B, and we could write big A, big B again, right? Because we have two big A's. And then we have big A, little b. Again, we could write big A, little b twice, but we don't need to. And notice, we only need 4 of these squares. So you're going to see lots of problems like this, and most of the time you're not going to be given the cross nice and neat. It's going to be a question where I'm going to ask you to use terms like homozygous and dominant and recessive, heterozygous, and you're going to have to be able to put the pieces together using the clues from the puzzle. Product rule, I'll go over this quickly for those of you who may benefit from this. So let's say you have a cross like this, and you're given a multiple choice question, something that you need to spend relatively little time on. And I ask you, what percentage of this would you find in this cross? Well, you could go and do that huge Punnett square and find it, or you could take each set of letters separately. Well, in the red one, if I cross those A's together, how often am I going to get big A, big A? 1/4 of the time. If I cross those B's together, how often am I going to get big B, little b? 1/2 the time. And so the chance that I'll get this out of this cross is 1 out of 8. Now, if that didn't make sense to you, that's fine. I can show you later. However, I'm more concerned that you can demonstrate this. And so if you can demonstrate those crosses, that's what I'm concerned with. If you can use the product rule, more power to you. All right, so some other instances that don't necessarily follow Mendelian genetics in the way that Gregor Mendel thought it was, and largely knew it was. One of those things is incomplete dominance. Incomplete dominance is when more than one allele is dominant. Remember I talked about how the dominant allele always shows itself. Well, if there's more than one dominant allele, in an incomplete dominant situation, there's incomplete dominance. Neither allele dominates the other one completely. And so look at this example here, where you have white, homozygous white flowers, which white is dominant, homozygous red flowers, red is dominant. When they mix together, neither one completely dominates the other, so you get pink. And so with incomplete dominance, neither allele expresses itself completely, resulting in an intermediate condition or a blending between the two. Again, flowers are a good example of this, or snapdragons in particular. And so, again, homozygous red, homozygous white, both dominant. What do you end up with? Well, you end up with broken up letters. I don't like the pink there. Here we go. Well, what happens if you mix red? What happens if you mix a pink with a pink? Well, kind of what you'd expect. Some of them are going to be red. Some of them are going to be white. and some of them are going to be pink. So that's incomplete dominance. Codominance, very similar idea. This is when you have more than one allele that's dominant. However, when they're combined, both alleles express themselves, resulting in both phenotypes being present. So, perfect example, these chickens here. You have a black chicken, you have a white chicken. Black and white are both dominant in chickens. And so when they mate, homozygous white, homozygous black, you have a 100% chance that you're going to have a checkered chicken that demonstrates both white and black. Not gray, which would be incomplete dominance, but checkered, both. So people often get incomplete dominance and codominance mixed up, so be careful with those. One, incomplete dominance, it's a blend. Codominance, both of them show, and there's usually like a mix, like a blending, or not a blending, a checkered or a stripe or something like that. Blood types are a great example of this in humans. A and B are both dominant. And so if an individual has both A and B type blood, that person has AB blood. There's not a blending, there's not a C type. They have AB blood. And so type O blood, so A and B are both dominant. Type O blood is actually recessive. So a person who has O is actually OO. Both the alleles are O. A person that has A can be AA or AO, right? Because A is or O is recessive. A person who has type B blood has BB or BO, and then a person who has AB obviously has AB. And so you'll see crosses with blood types as well. I'll pull Punnett Square here. Let's say someone with AB blood is crossed with someone who has A or who has AO blood. Well, we're going to make individuals all over the spectrum. They could have somebody with A, B, or AB. And so, again, very similar idea. Some other non-Mendelian traits are multiple alleles. This is when one gene has more than two allele possibilities. And so we've been dealing with just two allele possibilities, like short, tall. But there are other situations when a gene can have multiple alleles associated with it. And this usually results in like a hierarchy of dominance. So like here in rabbits, the wild type rabbit down here at the bottom is the most dominant. The albino rabbit here is the most recessive. And so in between, you have, you know, you have some different chinchilla and Himalayan and different colors that represent this hierarchy of dominance. Chinchilla is more dominant than Himalayan, but less dominant than wild type, and so forth. And so you have lots of different blends. This is very similar to human eye color as well. Some traits are called polygenic traits. This is when a trait has more than one gene that codes for it, usually resulting in a blending of phenotypes produced. So an example of this would be like human skin color. More than one gene for the trait. Human skin color has lots of genes associated with it, and so this is why you have lots of different skin colors, not just black and white, but you have lots of different tones in between. And then lastly is the concept of sex-linked genes. Now, sex-linked genes are genes that are only found on the X or Y chromosomes. Males have an X and a Y chromosome. Females have two X chromosomes. And so on the Y chromosome are found the genes for maleness. A Y chromosome allows you, look, when you're developing, that Y chromosome turns on and you become male. So most of the important genes that we're going to be talking about considering sex-linked genes are found on the X chromosome. An organism cannot survive without an X chromosome. And so recessive genes inherited on the X chromosome have a greater chance of expression in males because they only have one gene. And so let's look at this. So here's our X for females, XY for males. And typically the way that we'll write this is, let's say the gene for color blindness, for instance. Well, the gene for color blindness is recessive. So a female can have a dominant and recessive allele for color blindness and not have the disorder because she has two alleles, one of them being dominant, so she's normal. Whereas a male, all it takes is one recessive allele. Notice I'm not writing anything above the Y. Why? Because the Y chromosome doesn't have that gene on it. And so this is typically how we write when we're talking about crosses with sex-linked traits. We'll write the little B's and A's and T's and things above the X and Y to show this. So this individual will be colorblind. Here's a picture that if you can't see the 42 and the 74, you might be colorblind. And again, colorblindness is more prominent in males, but that doesn't mean that females can't have colorblindness. Hemophilia is another one. That's that a sex-linked disorder. Marfan syndrome is one. There's several sex-linked disorders that are associated with genes only found on the X chromosome. And so a sex-linked cross looks like this. In order to do this, it's a monohybrid cross between XX and XY. And so you'll perform that first. And so this is going to be the same. Doesn't matter where you put XX and XY. You can put them on the top, the bottom, whatever. And you're going to do this first. Just go ahead and do this. This is going to be the same on every single sex-linked cross that you perform. That's going to be the same. And so you're going to figure out which type of alleles are you given. Well, let's say that, for instance, there is a male that's colorblind. And so we're going to put a little b here because that male is colorblind. So he's the recessive allele because big B is normal and little b is recessive. Right? So he's going to have colorblindness. He has recessive. Well, let's say that his partner is heterozygous for the condition. And so now what do we do with this? How can we perform this cross? Remember, the Y chromosome doesn't carry anything. Don't put anything on the Y chromosome. Don't do it. It does not carry any genes. We'll never put a letter on it. Don't put anything on the Y chromosome. And so last, we're just simply going to perform the cross. What goes in this first square? A big B and the little b. What goes in this last square down here? A little b and a little b. What about the males? Dad's given the X chromosome to the boys, right? So he passes on, or the Y chromosome to the boys, so he passes on the Y. And so where do all males get their X chromosome from? Their mother. And so this big B is going to go here. This little b is going to go here. And so a cross between these two individuals would result in half the girls being colorblind, half the girls being carriers for the allele, meaning that they can give it away, but they don't have it, and half the boys being normal, and half the boys being colorblind. And so you can, usually when you list the genotype and phenotype percents for males and female in a sex-linked cross, you can go ahead and like put your male and your female symbol and kind of do those separately, like so, genotypes and phenotypes. All right, and so you'll have practice problems associated with these sex-linked crosses as well.